Hello, welcome to this Getting Started with CyVerse webinar. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory, New York. Welcome. This is the monthly webinar for our new account holders, and it's a live opportunity to get you started using some of the tools and services and features of the CyVerse platform. Over the next hour, I will give you first a brief introduction to CyVerse as a project, followed by a few hands-on demos. Before we actually get started, let me familiarize you with the technology that we're using in this Adobe Connect room. First of all, on the bottom of the screen is actually a place for you to ask questions, and I can monitor those questions off camera. That's really important. One of the reasons why we do this live is to give you an opportunity to ask any questions related to CyVerse and how to use it effectively. As you saw from the poll coming in, most people find out about CyVerse by word of mouth, so it's possible that you haven't had any actual training from us. The other thing is that along the side window there, you're going to see that there are a couple of downloads for you and a couple of important links. So you're able to download a copy of the presentation, and you can also take a look, and there are some guides that give you step-by-step tutorials of the things that I'm going to be doing or covering in these demos. So feel free to make use of those. If you look at the top of the screen in the corner, there is some arrows, a little button with 4 arrows in it, and that allows you to make the video full screen. That'll be important for later on when I do some demos from the computer. It'll make it easier to see. Finally, if you move your mouse over the screen itself, you'll be able to adjust the resolution of the streaming quality. So if you notice that your connection is not good, you can take the opportunity to switch to lower than HD connection. If at any time you have any kind of issues with sound or with whatever, please go ahead and send me a question. If you have any kind of computer issues, usually just leaving the room and coming back in will solve most of those. Okay, so let's get started with a little bit of an introduction to CyVerse as a project. And I have some slides that'll take us through that. So the vision of CyVerse is actually transforming science through data-driven discovery. And as a result of what we've been doing over the last several years, we have more than 40,000 users. We have petabytes of data that users store and analyze with us, and that's led to hundreds of publications, courses, and discoveries. The project itself actually started under a different name. Some of you may or may not be familiar, but it started in 2008 as the iPlant Collaborative, and it was tasked with providing these data-driven discovery, data-driven solutions for plant biology. The project launched its public tools in 2010, and it was successfully renewed in 2013. As we continue on, it was just last year in 2016 that we brought it adopted a broader scope and mission of transforming science to data-driven discovery. So really opening up to more than just plants, and that's what we continue to serve the entire life sciences community. One really important thing that we're quite proud of as a project is that we are entirely funded by the National Science Foundation. So we are your colleagues and your collaborators. We are not a business, we don't sell a product, but we're actually your colleagues. And this is a $100 million NSF investment, which provides these tools in a way that's really freely available to the community. So what that means for you as a user is that there are lots of resources here that you won't have to pay directly for a dime. And so we hope that you take advantage of as many of those resources as possible. Of course, we also ask that you think about citing us when you do make use of us, and also we are proud of the fact that everything is open to everyone. So whether you are a user located in the US or you're abroad, we are definitely fans of international and national collaboration. So I've used a term, and this is one that's very important to us. I won't dwell on it too long, but cyber infrastructure. What is that? Well, cyber infrastructure is data storage, software, high performance computing, and people. So things that you're already familiar with, I'm sure, but it's really organizing those resources into systems that help you solve problems of size and scope, problems that you might not be able to solve otherwise. Put simply, these platforms, tools, and data sets, those are things that you'll come to CyVerse and find available to you. You'll also find that we offer a variety of storage and compute solutions, as well as training and support, and today's webinar is just one example of that. Another key message that we want all of our users to take away is that CyVerse is built for data. So whether you're working with microbial data, plants, animals, any type of data, really CyVerse is built, and you'll find tools for all of those types of data sets. Some people, in fact, a large majority, are working with genomics data, but you may be working with images or other data types, and CyVerse is built for all of those things. So we really invite you to play around and look around. We won't have a chance to show you everything in 1 hour, but we'll at least give you an overview of what's possible, and please ask questions because oftentimes that's the best way to find out if we can help you with something. So this diagram gives you a quick overview of the CyVerse product stack. On the top layer are what we call the products. So these are the things that you as a user are most likely going to come to us and find. You access these over the web, whether it's cloud computing or web-based bioinformatics platforms. We have a variety of projects, and we're going to talk a little bit about them in the next few minutes, and then I'm going to go right to the demos. Beneath that are a whole set of services and capabilities which, depending on your level of use, most of us bench biologists, I think, are operating at this layer of just interfacing with these products. But if you are a developer or bioinformatician, you can fully customize the way that you consume these low-level CyVerse services. So whether you need to interact with our high-performance compute, databases, and storage, there are actually lots of custom ways in which you can interact with those, and we make those available to you as a user, mostly through APIs. When CyVerse was established back as iPlant, the community defined and continues to define our priorities. And they are still really driven by genomics, although not exclusively genomics. So when you come to CyVerse, you'll find tools already there for assembly, for RNA-seq, ChIP-seq, lots of different resources that you would need to do any type of high-throughput genomics analysis. Really, we use open source tools that are developed by the community. So things that you'd expect to use on your own or elsewhere, most likely they're already integrated into CyVerse and ready for you to use. Finally, it's important to recognize the different institutions that make up CyVerse. So the main corpus of people are at University of Arizona, along with the project PI. But then we have various people and systems spread out across the other 3 centers, including Texas Advanced Computing Center, Cold Spring Harbor Laboratory, where I'm at, and UNC Wilmington. We'd also recognize that there are increasingly different groups that are federating with CyVerse, and that includes the CyVerse UK project, which was a separately funded initiative funded by BBSRC in the UK. So now what I'm going to do is transition us into the demos, but first let me just quickly go through the products that we're going to want you to know about. We think of ourselves as building the pieces that you need to put together in a custom way. So you could maybe use the Lego blocks analogy, where we've crafted many of the solutions that you need, popular workflows, but you can also put things together in a way that's sort of freeform. At the probably first instance, almost every resource is going to make use of some type of data storage and sharing. And so we present to you the data store, which are the resources that you need to share and manage data with your lab, colleagues, and the community. Every CyVerse user has a 100 gigabyte allocation, but if you need more, there are terabyte allocations available. Those data are automatically backed up in 2 locations, and we've made it fairly easy to upload, download, and share, and I will be giving a hands-on demo of that in just a few moments. I'll also demo the discovery environment, which is a web interface to hundreds of bioinformatics applications. So these are bioinformatics applications, again, open source, that you'd normally run at the command line, perhaps on your own computer, but you'd be able to use them through this web interface in such a way that you don't need to actually use the command line in order to use them. It's also user extensible because you can add your own applications. Atmosphere will also give the final demo of the day, and that's our cloud computing resource. Whether or not you've used cloud computing, it's a really powerful tool for working in biology, certainly, and the other sciences as well. And it allows you access to hundreds of virtual machine images that you can fully customize. I'll describe that in case you haven't used cloud computing, may not be familiar with the terminology, but it's a really flexible and powerful resource that we make available to you. We won't give demos of the other products, but I just want to quickly mention them. The science APIs are science as a service platforms. And so if you are a bioinformatician or developer, these really allow you, again, to customize how you want to use the CyVerse resources. A DNA Subway is a wonderful tool for the classroom. So if you're teaching and you want to bring bioinformatics into the classroom, we have several bioinformatics friendly, or I would call them classroom friendly bioinformatics workflows that are streamlined and ready to use. Many of them support course-based research projects, and they've been quite successful in integrating bioinformatics and bench work to give students a really rich learning experience. Bisq is our tool for image analysis and management. You can work with hundreds of image formats, microscopic to macroscopic, and you can work with the Bisq APIs to develop your own image handling routines or use ones that are already integrated. Okay, so that was a very quick overview of some of the tools, products, and services of CyVerse, a little bit of background about the project. Now what I want to do is give you a few short demos on how to upload and share data, how to analyze over the web using the discovery environment, and also how to do an analysis in the cloud. So these are some really basic things that you want to get started with when using CyVerse. I'll remind you that at the bottom of the screen there are some guides. There's a discovery environment guide, a data store guide. So these are all links to the CyVerse Learning Center, where you can find some documentation. We're revamping that, so you'll be in the new Learning Center. We'll be following those links. And so on your own time after the webinar, you're certainly welcome to go through those, and they'll provide additional help in getting you started. This is also the time where you might want to make the video full screen if you have it. So up at the top, there is a little button there to make the video full screen, because I'll be doing most of this on my computer. Some things I can make larger and easier to see, some things I may not be able to, so you might want to make the video full screen. Okay, so getting data into CyVerse. As we mentioned, we have the CyVerse data store, which allows you to store any type of data that's related to your research. You can move those data seamlessly between different CyVerse platforms, and you can do automation, you can share. So let me just get you oriented here. There's three major ways that we provide for our users to interact with the data store. Two of them are point and click, and I'll demonstrate the CyberDuck software as well as the discovery environment. And then if you're familiar with the command line, it certainly gives you a lot of flexibility, and so the command line resources are there too. I'll demonstrate that as well. The discovery environment, which also is primarily an analysis interface, you do have some resources for uploading and downloading files, but it's rather limited since it's over the regular HTTP protocols of the web. So you can do it, but we recommend that you use CyberDuck for larger files. And so CyberDuck, it really almost works like Dropbox, where you have the ability to drag and drop folders. There's no size limit. We'd expect on a good connection you can move about 100 gigabytes of data within 30 minutes, and it's really easy to function on your desktop. Finally, the command line gives you the ability to script using iCommands, which are separately created binaries from the iRods project, which is the technology that underlies our CyVerse data store. So let me give you a little bit of a demo. Again, let me just make sure that you know that from all of these things, the links there actually go to learning, the CyVerse Learning Center. So I just want to emphasize that at the CyVerse Learning Center, the things that I'm about to show you can be found in this case, the data store guide. It'll be a rather full description of everything I'm going to cover, including where to download some things. So you'll need to, if you're using CyberDuck, you'll need to download CyberDuck. If you're using iCommands, the downloads are there. What I'm going to do now, of course, you're not expected to follow along with me. And I'll also mention that this is going to be recorded and posted to YouTube. So anything you miss, you're welcome to come back later and see in more detail. Okay, so let's actually talk about using CyberDuck. So to work with CyberDuck, the first thing you'll need to do is to download both the CyberDuck tool, which is not created by CyVerse, it's a third-party tool, and the connection profile. So you'll follow directions for those, and when you have it set up, let me just start CyberDuck on my computer here. Actually, I want to start a new window. I've got this one that's open and connected to my CyVerse data store. When you start it for the first time after you've configured it, there's a one-time configuration that should take literally a minute to do. You'll get this bookmark that will allow you to connect to the CyVerse data store. You double-click that. And then when you do connect, you'll see what I have here, which is the locations, the directories, the folders, the files located in my CyVerse data store. If I want to move any of those, I can simply take it and drag it to the location I'd like to move it. In this case, I'm dragging it onto my desktop, What will happen is that the CyberDuck tool will start a transfer window, and that gives you the status of the transfer. You can pause or restart uploads or downloads. You can obviously see the status and whether something is completed or not. Now, if you wanted to move a file from your local computer onto the CyVerse data store, that's really just as simple. I don't have a file on the data store, so I'm just, or rather my desktop, cleaned it up for the webinar, so I'm just going to create something. In fact, let's make sure we have that on the desktop. Okay, so this is an empty file, doesn't really have anything on it on the desktop, but I could just take that file and drag it over into the data store, and the same thing that would now occur, or that would be listed in my upload, and I could see that one is already completed. The other one is downloading. Of course, I am on wireless, so it takes a few seconds, but in any case, the transfer really is just as simple as dragging and dropping. And there's not much more to show you when it comes to doing a transfer with CyberDuck. There are more functionalities, but it really is drag and drop in order to move files. So the next thing I'll show you is iCommands, which is the command line version of this transfer capability. You would download iCommands here, and then you could follow the instructions that are located on the CyVerse data store to transfer using iCommands. There is a one-time configuration that you do at the beginning, but essentially that'll take a moment and then you're ready to work. So I'm just going to start a terminal on my computer, and I'll open a version that's easier to see. So to list the contents of my CyVerse data store, these are called iCommands, so they're very similar to Unix commands. I just, for the most part, put an I in front of them, so an ILS will list the contents of my CyVerse data store. Now, if I want to get a particular file or folder, in this case I'll get a folder, I can use the iget command. So I'll type in iget. I want a folder, so it's got to be recursive. I'll see the progress and be verbose, and I can get VCFs, and I can download that to my desktop and hit enter. And as you can see, that is already downloading, and there's the VCS folder that's on my desktop. Give it another second to finish. And you can double click on that, and then those files are there. And in fact, now it's done. So really moving a file or collection of folders, directories, whatever, are just a few simple commands that, of course, you can write scripts for. And that really makes it easy to move files from the CyVerse data store onto my local computer. I could also install iCommands on a server. If I have files, then I would, of course, want to skip downloading to a local computer and just transfer from one server into the CyVerse data store. Okay, so the next thing I want to show you is transferring or working with data, really sharing data in the CyVerse discovery environment. So to do that, I want to go to CyVerse.org. This is the home page for CyVerse, and I'm going to go ahead and launch the CyVerse discovery environment. As you may have seen, if you did go to the Learning Center, there is a discovery environment guide, which will take you through anything that I'm going to show you here. So feel free at your leisure to come back to the discovery environment guide, and it'll go through these very basic capabilities. Okay, but from the discovery environment on the CyVerse homepage, I'm just going to go ahead and click Launch the DE. And let's see, oops, I want to go actually to the new link. And I'm going to go ahead and log into that. Okay, so this is the CyVerse discovery environment that's loading here. And it opens up the last things I was working on, so I actually want to close some of those. I don't need them right now. And this one too. Okay, so when you come to the CyVerse discovery environment and you're ready to work with some data, you'll click on the data window. And if you click on your username, which is the name of your home folder, you'll see all those things that I saw a few moments ago, both in CyberDuck as well as the iCommands. If I want to download a file, I do have the ability to go here and I can click download and download a file. I can upload, I can even import from a URL, so some basic capabilities. What I want to actually concentrate on is sharing data because the Discovery environment is a really wonderful place for working with sharing data and managing your data and even your metadata. So to do that, I might select an individual file that I want to share, and then I can click on share and say create public link. I click on that file again and say create, and it's going to give me a URL. I can go ahead and show that link and copy it, and then if I put that link into any web browser and hit enter, it will start a dialog that will ask me to go ahead and download that. So I could give this link, this URL, to anybody who I wanted to download a particular file. And so it's a really easy way, again, mirroring maybe Dropbox, where you can create a quick link to share a file. You have to be aware that this does not work with folders, so that's a limitation here. We'll talk about sharing folders in a moment, but sharing files, this works perfectly well. It also reminds you that when you're sharing files this way, this really works well for smaller files. I wouldn't suggest sharing multi-gigabyte files this way because it is really quite slow compared to the transfer speeds that you can get on CyberDuck, or certainly the way that we're going to show you sharing with a CyVerse account. This link is called a public link because anybody who has that link can use it. So if you want to delete the link, then you can come back to the same window and click the little deletion icon, and that will delete the link. And if someone uses that link again, now they'll get an error that it's not found. So it does allow you a little bit of flexibility and privacy, but you want to use these links for things that you probably don't consider too sensitive and that aren't too large. What if, though, you want to share a larger collection of data? So in this case, we recommend that you share using the Discovery environment, and this will require the other person to have a CyVerse user account, but it's free and it doesn't cost them anything, and it's not too difficult. So in that case, you can select a folder that you want to share them with, and then you click share, and this time say share with collaborators. And then sharing is as simple as now finding the name of the person you want to share with. In this case, I'll put in the name of a colleague, and I'll get their name as well as their institutional affiliation. I can click enter, and then I can decide what level of sharing I'd like to give them. Now going back to the guide here, there's an explanation of what the sharing terms mean. So if you want to share data with another user, these permissions are based on the Linux permissions. So giving them read access allows them to read and download the file and to view the metadata. Write, they can edit the metadata or edit that file. And then the highest level of permission is own, where they can do everything. And so you have to be careful. You don't want to give own permission and they accidentally move or delete that file. We wouldn't be able to help you very much if that's the case. So decide the appropriate level of permission, and then you would click done. Now I'm not going to do that here, but if you did click done, you'd get a notification that that file is being shared up in the notifications window that's up there. When you do go to share a file, and I'm going to open that window again, if something is shared with somebody already, then you would come back to the same file, and then if you have one or more people you've shared it with, if you want to revoke those permissions, you can simply click the X next to their name and click done again, and then those permissions would be changed accordingly to what you selected. When you share a file, no matter what the size, literally within a few seconds, the person you shared it with has access to it, and it'll be available in the Shared With Me folder that is located on the left-hand side. There are several folders that everyone has something shared with them by various services or users, but you'd also find that person here. You could also go to the notifications, and anything that's shared with you, you click on the notification and it will navigate directly to the location of the shared file. So that's, well, one more thing I could show you really quickly. I do want to talk about metadata, although I don't have exactly all the time I'd like. I'd like to point out that when you do have data here, and let me just bring up an example of one that I've, well, I have one here. If you want to look at an example of this, one of the great things about CyVerse is that everything is really built for metadata as far as our data are concerned. So when you have a folder, you can only name a folder with so many underscores before it just doesn't make sense anymore. But if you go and do something like add metadata, Well, I could click on Edit and View Metadata, and I can see that this particular folder has a whole range of metadata attached to it. In this case, these are metadata that are associated with a submission to the SRA using CyVerse's SRA submission pipeline. But viewing this metadata, these are the metadata that are needed by the SRA to appropriately submit a sample for archiving into NCBI. I can search everything in CyVerse using just this sort of simple search. But if I spell it right, I'll get better. But if I use the advanced search with this little funnel here, I can actually search according to metadata attributes, values, and units. So for example, if I had the value genus as a metadata attribute, and I had Dioscorea as the value I'd want to pull back, instead of getting all those irrelevant results, or potentially relevant results, or just more results than I want to look at, I can exactly search for things according to whatever metadata that I have put in. And you can use an arbitrary metadata template, or you can actually assign metadata according to any of the variety of templates that we provide to you. And those are in metadata download templates. You'll see a variety of the ones that are there. If there's something you need there and you think is pretty commonly used, we'll also support that too. It's taking a while to search since I think I'm on wireless. That'll eventually come up. But in the meantime, let me introduce you to the other capabilities of the discovery environment. And I'll go back. I have just a few slides to orient us on that. Okay, so moving into the discovery environment, if we want to analyze our data, the discovery environment, as I mentioned, is a platform. So it gives you the chance to run almost any bioinformatics application. It's seamlessly integrated with all of the other services, so you can use the data store at one moment, and then those files will be available for you in the discovery environment. It's also user extensible, although I won't have a chance to show you today for lack of time. You can add your own applications. For anybody that is a bioinformatician or developer, those applications are all Dockerized. So we really make it fairly easy for you to take an application that exists elsewhere and move it onto the CyVerse system and use our resources to execute a tool or workflow. We've already sort of covered the data window, so I won't go too much into that, but there are two other sort of panels that we're going to talk more about and actually use. They are applications or apps. So in the apps window, you have the ability to execute an application, to build a workflow, or to integrate new applications. And in analyses, you can see the results of a job, monitor the status of a job, and you have a detailed history of jobs that you've executed. So I'll do a quick demo, and in this demo, we're just going to take unaligned DNA sequences that are in FASTA format, and we're going to create a multiple alignment. So very simple application, but one that gives you a chance to see all the bits and pieces. Okay, so let's go back to the discovery environment. Okay, I had a problem with my search there. And from here, we will do a multiple alignment. I will go ahead and click on Applications. And in this case, the application that we're going to use is one called Muscle, so I'll just search for it, although the applications are listed by topic or operation. When I find the Muscle application, I know it happens to be one of the ones that I favorited. You could see it has a rating of some stars. If I want to know more about the application, I can click the Info button, and that will give me a little bit of information about the application. To actually use the app, I can click on the name of the app, and it's going to load. And I'll take that other menu out of the way. So all the applications within the discovery environment look very much like this, where you have this sort of accordion menu of options. On the first page, you have the ability to list some comments. So I'm just going to write, this is a demo webinar. And what we're going to do is select some input data. So I'm going to browse. And it browses to the last location you've been in. I, however, am going to take some data. This is the same that's in the guide. I should make it a favorite. That would have saved me some time. You can certainly do that, but that's in CyVerse training. And we are working with the discovery environment. And we're going to go ahead and use the muscle input. All right, so we're going to use this FASTA file. This happens to be a DNA sequence, so I'm going to set that option. And then I click Launch Analysis. When I click Launch Analysis, I'll get a notification that the analysis is launched. And then I can check the status of the analysis in the Analyses menu. So right now, the analysis is running. And I can do various things. If I didn't want to run that analysis or I realized there was a mistake, I could go ahead and click on cancel. But for right now, I selected analysis and I want to go ahead and view the parameters. So really important, especially a little bit difficult when you're working with a point-and-click interface. You do have the option within the Discovery environment to actually see the previous jobs you've launched, as well as any parameters associated with those jobs. So in this case, I can see all the parameters associated with this job, including the actual files that I used as input. In fact, clicking on the name of the file there will allow me to navigate to that file in the Discovery environment, so I can open it. This really helps me because sometimes you have files that are just not human readable names, lots of letters or numbers, alphanumeric sequences for the naming schemes. So this is really nice. And I can see any of the settings. For an application that is more complicated, this starts to make a difference. Let's see if something that I have used that has a lot of options, something like Top Hat. And if I go to that and I view the parameters, I can see all of those parameters. So this makes it easy to be reproducible. If I click Save As, I can actually save these parameters as a text file and location of my choice. The other thing that's a nice feature here is that I can also take an analysis, and let's say I want to do it again, I can click on that analysis and click Relaunch. And then rather than filling in every single option or every single file that I did before, the application will launch in a window with all of the settings and parameters that I had previously chose. And then if I just want to adjust one, if I wanted to make this auto instead of DNA, I could launch the analysis without having to go back and set. In this case, it's only one parameter, the input, but maybe I had 10 or 20 that I didn't want to reset. So you do have the option to reanalyze. If I click refresh, I can see what the status of this job is. It's still taking a moment to run. I'm not going to wait for it. It should finish any second now, but I'm just going to click on one. When a job is finished, you can click on the name of the job, and then it will automatically direct you to the output of that job. In this case, the job outputs some aligned FASTA sequences. Okay, so this is my aligned sequences, and I can see those there. So the analysis window really gives you a complete look at your history, things that you've done previously. One other feature that's important here is you can share an analysis with a collaborator just as you share something, a file. This allows you to share the results as well as the input files with someone else who's working with you in the analysis. It also can be helpful for debugging, so if you're working, you may want to share an analysis with the CyVerse support staff in case you need help in doing something. Okay, so that is a really quick overview into the Discovery environment. You have the ability to certainly access data that you've uploaded. Then you can go into the Applications menu. There's several hundred bioinformatics applications for everything from RNA-seq to genome assembly and more. You can choose the appropriate applications from there, and then you can launch those. Once you've launched them, you go to the Analyses window, and you can see the results, you can see the status, and the history of things you've done previously. At this point, I also want to remind folks, if you have any questions, please at the bottom of the screen go ahead and type those to me. I haven't seen any so far, so hopefully that means I'm doing an okay job explaining things to you. But if you do have a question, let us know. And we've got a first question. So let me just go take a look at this. There's a person who's working and asking, they've got their own cluster. Let me actually adjust this so I can see the whole question. Okay, so the question is, you're at a research computing cluster, and can you run something from your university's computing cluster, or do you have to run it through our portal? And the answer is yes, you can run it through your own computing cluster. So that is not a everyday thing that people ask us. The majority of our users are just looking to use their own tools, but you can actually do what we call federation with CyVerse, which means that you can add, or you can register your cluster as a resource that can be used by CyVerse. And then you can also designate who is allowed to use that resource. So say for example, you have 10 users in your department. You want to provide them the ability to launch a certain workflow. They would like to use it in the Discovery environment because it's easy to use. but you want it to run on your own system. Maybe the data is sensitive, or maybe you want to be first priority and not have to wait for queues. A number of reasons. You can do that using the Agave API. So I won't have a chance perhaps to give you a very, very detailed answer, but if you contact us, support@CyVerse.org, we can walk you through all of the steps and point you in the direction of documentation. So I'm looking to see, I don't see a follow-up question yet, but if you do have a follow-up question to that, please let me know, and I'll certainly answer that follow-up question as well. But hopefully that answered some of what you were looking to hear. Okay, so I have time now for one more demo, which is the cloud computing resource called Atmosphere. So let me just bring up a few slides I have to orient us. Okay, so Atmosphere. So Atmosphere is a really easy to use, we think, cloud that allows you to have access to hundreds of virtual machine images. It's a fully customizable way to work with your software, and it's integrated again with our other data and computing resources. First, I should explain just two technical pieces or piece of jargon vocabulary concerning cloud computing. The first is the concept of something called an image. What is an image? Well, let me use an analogy. I'm sure most of you have bought a computer and you have an old computer and you want to take files from the old computer and put it onto the new computer. So in order to do that, you take the original system, you copy those files or documents onto a backup hard drive. But there's another type of file that you could make, and that file is called an image file. An image file is a complete clone of the original system. It has not just individual documents, but it has the operating system, it has all of your settings and your parameters, everything on one file called an image. Now, if you take files or documents off that backup hard drive, there's still those files or documents. But if you take an image, you actually can restore your original computer down to the graphic on your desktop. So that's what an image file is, the complete system, including the operating system, any installed software, any data sets, any of those things would be on an image file. The other concept or vocabulary term is called instance. So in the CyVerse cloud, we have a variety of physical resources. We have hard drives, CPUs, memory, RAM. And so when you come to CyVerse, you're asking us for an allocation of some of those to use temporarily. So you tell us how much we need and we assign that to you. Also, since essentially you're building a computer, you need an operating system, so you can pick an image that's configured to your needs. And when you put those both together, what you end up with is a virtual machine. And that virtual machine will be available to you at an IP address, and that's how you'll access it. The Atmosphere service allows you to choose existing images or customize them. You can get instances of up to 16 cores or 128 gigs of RAM. You can access them via the shell or via VNC, and you can also share your images because you're allowed to image your own resources with other users, or you can make them public. There are a lot of use cases for this because essentially what Atmosphere is giving you a on-demand computer. You ask us for it, and within a few minutes you have a fully customized computer that you can use for any purpose. Some interesting use cases are running some of the software that is currently monopolizing your laptop. You can also run some programs that already have their own graphical interfaces. Those wouldn't integrate into the Discovery environment. You have full administrative access or pseudo access, so you can install at will. You don't need permission from anyone. It's a really nice way to make a uniform computing setup. So if you want to be reproducible in your classroom or with your collaborators, you can definitely use a virtual machine image. And it's also a way to make your own software available to a larger community by placing things on an image that everyone can use without having to worry about how to configure or install. So I'm going to give you a quick demo on how to launch an image and connect to one, or launch an instance and connect to one. There are a variety of ways to do that. You can download VNC Viewer if it has a desktop. You can use a shell or PuTTY, whatever type of way for accessing the terminal. We have a really cool way of doing it straight through the web, so I'm going to show you the web interface. So let's go to Atmosphere. I'm going to close this. And remember, there is in the CyVerse Learning Center a Atmosphere guide, but I'm going to go ahead from the CyVerse homepage. and click on Atmosphere. And from here I can click Launch Atmosphere. Okay, now I see a variety of images. These all have software installed. I could search for various things, like if I was interested in RNA-seq, I could see the types of tools that are installed in a particular image. First of all, I'm going to log in, and I think I'm already logged in with the Discovery environment, so I'm good. I will remind you that you do need to get, somebody's replying to me because we do have live help chat, you do get a or have to apply for Atmosphere access once you get your CyVerse account. So it's not automatic. You'll have to go back to user.cyverse to request access to CyVerse. That's described in the Atmosphere guide. Once you do have access, you can come and see what you're currently using, what you've used in the past. In this case, I'm going to say I want to launch a new instance. In this case, I'll launch an instance that is basically blank. So these are base images. They don't have very much installed. So I'll click one of those. I can see some descriptions about it, but I can go ahead and click Launch. And then I can decide on what cloud and then how much I need in terms of resources. Now, I will warn you that not every single instance size is available every single moment of the day. If there's heavy usage, then some instance sizes might not be available if we're at capacity for that size. Also, when you come to Atmosphere, you'll have a certain number of what we call allocation or Atmosphere units. Those run, they give you an initial allocation, and every month that allocation refreshes. So if you over capacity, you can request more resources, but if you are launching something, you have to be mindful of this. And the purpose of that is just not to waste resources. We don't want people launching 20 instances and then they don't come back and use them. So to launch this one, which I'm not going to do, you click Launch Instance. When you do click Launch Instance in your project, you would see that you have an instance. It would take a few minutes to get this green active state, but once it's listed as active, it has an IP address and you're ready to connect to it. If I click on the name of the instance, I get some more details, and if I want to connect to it, I can do that right in the web, in this browser. So if I open the web shell beta, I'm going to have to enter my CyVerse password. Try to make this, whoops, should have not done that. There we go. There we are. So now I am actually in CyVerse or on this instance at the command line. And I could see, my directory, I could make a directory, but I'm actually connected via terminal from the web on that Atmosphere instance. If I go back and I want to see the desktop, since this one has a desktop, I could click on this link. And now I'm on the desktop of this remote machine. I could go ahead and open a folder. I could, open a web browser within there, anything that I would do on a normal computer. Of course, I can connect from my own computer. So on here, I need to go back to Atmosphere and get my IP address. So that is here, and copy it. And then I can use SSH, my CyVerse username at that IP address. I can say yes to connect and enter my CyVerse password. And now I'm connected again. If I go back in my desktop, and let's say from here I want to make a directory, that directory is created on my remote machine. So any bioinformatics application I install, I can use. And so this is essentially, an on-demand computer. I'm sure you've been in the experience where you needed to run a certain software, and maybe it had dependencies, maybe it required a certain amount of RAM or disk space. Some of these systems, if you look as configured, could be tens of thousands of dollars to buy off the shelf. But within a few minutes, you can have that machine available for you to use. You would then, the next steps would be to transfer data from your CyVerse data store into your Atmosphere instance and then analyze your data. When you're done, you could transfer it back into the CyVerse data store, or you could transfer it into a local volume that exists within Atmosphere, and these things are described in the guide. So now back to what do you do when you're finished. Let me go back to Atmosphere. So when you are done, there's a couple of options for you. One, if you are just done for the moment, you might stop or suspend your instance. If you've installed something and you want to be able to launch an instance that's now been configured by you in the future, you might request to image that instance, and then it would appear in the catalog. If we're truly done, then you would click delete. Now, you have to be careful because once you delete an instance, there's no one in CyVerse who can help you recover any information. So you want to make sure that you move anything off that instance first. I'm not going to delete it just in case there are questions, but you get the idea that you can delete that instance, and then you'd be able to have your allocation back in terms of the numbers of CPUs that you're allowed to launch, for example. But this is a really useful service because you're able to really on demand just launch whatever you like. So I have one more slide, just sort of wrapping up and talking about what to do next. I'll remind everybody again, if you have questions, please at the bottom of the screen send me the questions and I'm more than happy to answer. Okay, so coming back here, what are some of the next steps? We've showed you the discovery environment, we've showed you how to move data, and we showed you Atmosphere, so what's next? Well, one thing that we encourage you to do, and let me just pull up the last slide there, is to take a look at the CyVerse website, where you'll find links to the Learning Center, you'll find links to tutorials and videos and documentation, as well as upcoming workshops and webinars. And then I want to finally finish the slide deck and return to the question I have by acknowledging the executive team across all of the different sites.